Hey guys, welcome back to One Stop Biology. And today we are going to start with the post-fertilization structure and events. And we are going to study structure of two different events. One is endosperm and the other one is embryo. So till now we have covered almost the entire chapter 2, which is sexual reproduction in flowering plants, which is NCERT class 12th chapter 2. Okay. So till now we have studied the pre-fertilization events, which is formation of male and female gametes. How does the structure of a male and female gamete looks? How does the male and female reproductive part looks? How does it occur? And then in the last video we studied how does the pollen pistil interaction occurs, and then how do we achieve double fertilization and what exactly happens in double fertilization. How does an endosperm and an embryo is formed from what? From the primary endosperm cell and zygote respectively. Okay, so let's see what happens in post-fertilization. Now, in post-fertilization, basically there are several events. So first is endosperm development. Second is embryo development. Then there is maturation of ovules into what? Seeds. And maturation of ovary into fruit. Okay, so these four events collectively asked are known as post-fertilization events. So today we are going to cover the first two, which is endosperm development and embryo development. And then in the next video, which would be the last one for this chapter, we are going to cover maturation of ovules into seed and maturation of ovary into fruits. Okay, so let's see what exactly is an endosperm. So endosperm development, endosperm is formed from what? It is formed from the primary endosperm cell, which is also known as peck. Okay, so the endosperm is formed from peck, the primary endosperm cell. So now endosperm development precedes embryo development. So before the embryo develops, endosperm development occurs. Now, why does this happen? Because the primary endosperm divides repeatedly and forms a triploid endosperm tissue. What happens is the primary endosperm cell divides repeatedly and forms a tri triploid endosperm tissue. Now, the cells of this tissue are filled with reserve food material, and these are required for nutrition of the developing embryo. And that is why always endosperm development occurs before embryo development. Okay, so in most common type of endosperm development, the pen, which is primary endosperm nucleus. So the primary endosperm nucleus undergoes successive nuclear divisions and it gives rise to free nuclei. Okay, so this pen gives rise to free nuclei. Now, this stage of endosperm development is known as nothing but free nuclear development. Okay, so this is free nuclear development wherein free nuclear endosperm is occurring. Okay, so subsequently what happens is cell wall is formed on these nucleus, basically on these free nuclei. So the cell wall formation occurs and the endosperm becomes cellular. Okay, now the number of free nuclei formed before the cellularization, that varies greatly, As if we take an example of coconut, the water inside the coconut that you see is nothing but the free nuclear endosperm. And the white kernel wala part, the white tender coconut wala part is nothing but the cellular endosperm. So that is formed after cellularization of these free nuclei. Okay. Now these endosperm may either be consumed completely during the embryo development, as in that happens in the case of peas, groundnut, or beans, where you don't find any kind of endosperm, right? You just find the seed inside. So endosperm can be completely consumed in the developing embryo, and sometimes it is not completely consumed in the by the embryo, as in the case of coconut or castor. Okay, so it persists till the seed matures. Okay, and it is used during seed germination. Okay, so it is later used during seed germination. This is all about development of endosperm. Now, what happens in embryo development? Let's see that. Okay, so the embryo that forms from the zygote, right? So the zygote is developed into an embryo, right? So this embryo develops at the micropylar end of embryo sac, right? Where the zygote is situated, right? Now most zygote divide only after certain amount of endosperm is formed, and this is because it the endosperm which is formed that provides nutrition to developing embryo. Okay. Now though the seeds differ greatly, early stages of embryo development, which is known as embryogeny, is very similar in monocotyledons and dicotyledons. Okay, so let's see some stages in embryo development. So here, this is the zygote. Okay, now this becomes two-celled and it starts then dividing into various cells. Now it can be a globular embryo. It can also be a heart-shaped embryo. Okay, these are known as suspensors of an embryo. Now, here again, you'll see that the embryo is more developed, and you can see radicle and plumule, from which the stem and root develops, and these are the cotyledons. Here, since here you can see that there are two cotyledons, this is embryo development in a dicot. This is embryo development in dicots, dicotyledon plants. Okay, now what happens in a typical dicotyledonous embryo? Let's see that as well. I'll just clear my screen and then we'll see what happens in a dicot embryo. Okay, so a typical dicotyledonous embryo consists of an embryonal axis and two cotyledons, right? Now the portion of embryonal axis above the level of cotyledon is the apicotyle, and that terminates with the plumule, or from there germinates the or develops the stem. Okay, so that is the stem tip. Now the cylindrical portion below the level of cotyledon. Okay, so the cylindrical portion below the level of cotyledon is known as hypocotyle, and that terminates at its lower end in the radicle or root tip. Now the root tip is always covered by a root cap, and this we have studied during the topic of root as well, right? So here if you see the diagram, basically these two are the cotyledons, right? Now the part above the cotyledon is plumule, which forms the stem, And the part below the cotyledon, this cylindrical part is hypocotyle, and the part above is known as epicotyle. Okay, so this forms the radicle from which forms the root, and root tip is always covered with root cap. Okay, so this is the diagram of a typical dicot embryo. Okay, now what happens in the embryo of monocotyledons? Let's see that as well. So in case of monocotyledons, in a monocot embryo that possess just one cotyledon, right? Now in the grass family, the cotyledon is known as scutellum. Okay, and this is situated towards one side of the embryonal axis, which is known, which is the lateral side, right? And towards the lower end, the embryonal axis has what? It has radicle and the root cap. Okay, so basically that is known as coleorhiza. Okay, now the portion of the embryonal axis above the level of attachment of scutellum is the epicotyle. And epicotyle has what? Basically, it has the shoot apex and few leaf primordia. Okay, and that is enclosed in a hollow foliar structure which is known as coleoptile, right? So let's see the diagram of the embryo of a grass. Here, if you see, there is the towards the lateral end we have scutellum, which is nothing but the cotyledon. Okay, this is nothing but the one cotyledon. Then we have the coleoptile, which forms the stem and leaf apex. So basically, this is the shoot apex. Here we have the epiblast. Now towards the other end, we have radicle, we have the root cap, and this is the coleorhiza, right? So this is the structure of the embryo of a monocot. And do you remember what exactly coleorhiza is? So basically the radicle and root cap are enclosed in an undifferentiated sheath, and that sheath is known as coleorhiza. Okay, so guys, with this we have finished the first two events of post-fertilization, which is development of an endosperm and development of an embryo. And within embryo, we have covered both developed development of a dicot embryo and development of a monocot embryo. Now, in the very last video, in the next video, we are going to cover seed and fruit, which are the next two events of post-fertilization. So I hope you understood the two events that I have discussed in this video. If not, please drop a message either on the comment section of the video or over WhatsApp, and number is given in the description of this video. And if you did understood the entire video, if you did understood all the topics that I have covered in this video, please do not forget to like the video and share it with your friends as much as you can. And yes, guys, please do not forget to subscribe to the channel. Thanks, guys. Bye bye.